This is a very interesting paper that looks at COVID viral entry and replication in the nasal passageway, where the general flow is that COVID will enter through the nasal passageway. It will bind to ACE2 receptors on the motile cilia, and then it will inject the viral RNA after fusing to the motile cilia. These are like little windshield wipers that exist in the nasal passageway to sort of move things along. And then after they bind and inject their viral RNA, they actually reprogram the microvilli, causing them to grow rapidly and then bud with more virus particles, and then these will be shed into the mucus, which will then flow laterally and infect neighboring cilia and repeat the process. Where after this initially happens, it causes those local microvilli to grow, and then viral particles bud and get ejected into the mucus, which then flows laterally from this windshield wiper effect of the motocilia, causing infection of neighboring cells, which expands this growth. And this happens quite quickly, where from 6 hours after contact, you have a little bit, 24 hours, you have a lot, and then by 48 hours, you have significant levels of growth and viral particle ejection. Now, diving more into the study used nasal epithelial organoid cultures. So this is an in vitro study, a lab study. So in vivo is in life, in vitro is in culture, where they found that COVID attaches to the motile cilia using the ACE2 receptor, and that Omicron variants bind with higher affinity to these motile cilia and show accelerated viral entry. So in culture, what does this look like? So within 24 hours, the green being the spike protein, you have a little bit of binding, and then all of a sudden by 48 hours, you have an explosion everywhere of it. And that this infection is a two-step process, where first COVID binds to the ACE2 receptor on the surface of the cilia, the motile cilia, and then within the next 24 hours, COVID hijacks the host cell machinery to induce elongated and highly branched microvilli, the growth of the microvilli, where the COVID particles then bud and are spread laterally, transported to neighboring microvilli and cilia. And while ACE2 is the major receptor that it binds to with the spike protein, there's also the TMPRSS2. And then using electron microscopy on these motile cilia, we can see the COVID particles themselves, which are these tiny little beads. And then this is what that viral fusion it looks like for the COVID particle to the motile cilia, where it fuses to the membrane of it and then injects the viral RNA. And then here are images of the motile cilia, these long stalks with the elongated microvilli. And again, all these changes occur within the first 48 hours of infection, with changes to the microvilli occurring as early as 24 hours after initial infection.